Hi, welcome to day 3 of the California Seaweed Festival. Our theme today is Seaweed Science. I'm Dr. Janet Kubler, co-organizer of the festival and moderator for this session. I'm a marine biologist currently working at California State University at Northridge and University of Connecticut. And I have been using science to learn more about seaweeds for more than 30 years. Oops, I have to get rid of something. California has a rich history of scientific research on seaweeds, and currently hundreds of students and professional scientists are engaged in learning more about seaweeds with science. So I wish we could highlight everybody. I wish we could bring everybody in and talk about seaweed science. But in this hour, we are excited to bring you a panel of 4 scientists working in diverse systems using a wide range of different techniques to illuminate our understanding of seaweeds. I will introduce them one at a time. They'll each have an opportunity to tell us for 5 or 10 minutes about their work. And then after we hear from all the panelists, we'll be open to questions. So please put your questions for the panelists in the chat on the YouTube live stream. Okay, panelists, ready to go? Okay. All right, first up, we have Dr. Jennifer Smith, who is Associate Professor of Marine Ecology and Conservation at Scripps Institute of Oceanography at University of California at San Diego. She has great California seaweed credentials. She's a graduate of Humboldt State University in Arcata. She received her PhD at the University of Hawaii, where she was mentored by Isabella Abbott and Celia Smith. And she was a postdoctoral scholar at National Center for Ecological Analysis and Synthesis at University of California, Santa Barbara. She's an expert on Pacific seaweeds and coral reef ecology and has published almost 100 peer-reviewed articles. She's mentored more than 20 graduate students and more than 120 undergraduate students. And her research focuses on understanding how benthic marine communities are structured and how they function through natural and anthropogenic changes. And a lot of time, it seems like that hinges on what the seaweeds are doing. And one thing I really have to share is, when I asked Dr. Smith to tell me how, something about herself that she'd like to make, have highlighted here. She wanted to highlight that she's a phycophile, which literally means seaweed lover. And I think you're in good company here. So please, Jen, tell us some about your work.
Absolutely. Thank you guys so much for this opportunity. And thank you to both of you, Simona and Janet, for organizing this event. It's long overdue, and I think all of the phycophiles in California and beyond are much grateful for the opportunity to be involved, and we hope that this continues for many years ahead, every year, and hopefully we can be in person sharing our seaweed stories and seaweed recipes together in the future, hopefully next year. As Janet said, I'm going to share my screen and share a little bit about my lab. So as Janet said, I'm a marine ecologist, and my lab, I have a diverse lab group. All of us, I would like to say, are phycophiles. The team can speak for themselves, but I think I've convinced most of them that seaweeds are an incredibly important and often vastly underappreciated component of marine ecosystems. My lab team studies marine ecosystems in a variety of different habitats. We focus on both coral reef ecosystems as well as California's coastal ecosystems, working in both the rocky intertidal and the kelp forest systems. I have a great team of students, and I'd like to thank them all for the work that they do and for the inspiration that they continue to give me every day. the focus of our work is largely to understand how different types of environmental influences affect the structure of marine communities. So these can be broken down into things like local factors, whether you want to think about the introduction of non-native species, such as this new invasive Dictyota pictured in the top left, over-harvesting, fishing, the introduction of pollutants, or any other type of local factor that might be influencing community structure, as well as global factors. So things like climate change, sea level rise, rises in sea surface temperature, and ocean acidification. All of these things are happening to our marine ecosystems here in California, and understanding how these factors affect competitive interactions between the organisms that live in these coastal ecosystems is highly important for understanding how these communities are structured and how they're likely to change over time. And so this is a large focus. I mean, it's a very diverse area of study, and it's these are all types of topics that my students and myself are currently working on here in California. Just to give you a few examples of some of the specific projects that we're working on, particularly excited by this project, which is known as TIDES, which stands for Tribal Intertidal Digital Ecological Surveys, where we are using large area imaging, where we take thousands of pictures of the rocky intertidal and we stitch them together into a 3D photo mosaic, where you can basically go on a virtual exploration of the intertidal zone as if you were a bird or an insect hopping around this intertidal. And what this does is it preserves these ecosystems in space. It allows us to study the distribution of a whole variety of seaweeds as well as invertebrates, and how they change over time, because we can survey the exact same places, and we're able to collect so much data by using this large area imaging approach. As we all know, we're often getting chased out of the intertidal by the tides, and so by being able to literally bring the tides, the intertidal home with us, we're able to learn a lot more than we have been in the past. And this project is in partnership with some indigenous nations, specifically the Tolowa Dini Nation, which is up in Smith River, which is in the very farthest north part of California, where they have incorporated this approach in their traditional monitoring of the rocky intertidal to study important traditional species that they have utilized for thousands of years, including Pyropia, otherwise known as Porphyra, or in their native language, Lot, which is very abundant up there. So this is a really fun and exciting project. I'd like to thank my partners at the Tolowa for their effort in this work. Another project that I'm perhaps the most excited about right now is using seaweeds or products from the sea to solve some really important climate change issues, specifically reducing greenhouse gas emissions. So some of you may have heard of this new innovation, but basically this specific species of red seaweed, Asparagopsis taxiformis, which exists here in Southern California, has been shown that when fed in very small quantities to livestock can prevent methane emissions or reduce methane emissions by up to 90%. Methane is a really bad greenhouse gas. It's about 30 times more potent than carbon dioxide, but up until this seaweed solution, there hasn't really been any way for the livestock industry to become more green, at least in a significant way. And so this is an example of where seaweeds can literally have a major impact on a completely different industry, that of terrestrial farming and livestock production, to ultimately reduce greenhouse gas emissions, to ultimately, hopefully, get this climate crisis under control. So we're working to try to understand how to cultivate and allow this species to be commercially viable, which is a lot bigger of a challenge than it may sound like initially. So we've been working with Asparagopsis in my lab now for the last 2 years and have made great strides. I have a whole team of students and postdocs that are working on this, and we continue to try to unlock this tricky little beast, that being Asparagopsis. I'm also really excited about studying seaweeds that have important culinary applications. We know seaweeds are a superfood, order of magnitude more nutritious in terms of vitamin and mineral production than any terrestrial crop that we can grow. Yet they are highly underutilized. I think, as you guys have heard on various panels throughout this festival so far, There are a lot of small businesses that are emerging that are using and utilizing and growing California seaweeds, but still the majority of seaweeds consumed in the US is imported or are imported from Asia. And so we need to continue to advocate, to study, to optimize growth of California seaweeds and to get them into our market fresh and edible. And so I'm happy to support students like Branch Adlowski, who started California Seaweed Company, and many others of you out there. I'm a fan of yours as well. So I'd just like to end by saying that I have the great honor of being a student of a wonderful team of female scientists, specifically female phycologists, including Dr. Izzy Abbott, who is a legend in her own time and literally was the founding mother of the field of phycology in many ways. She's described hundreds of species and has many more named after her. And I was fortunate enough to be right down the hall from her when I was doing my PhD and learned seaweed taxonomy from her. And it was unlike anything I can ever wish to do. It's just an amazing experience, as well as mentors such as Celia Smith and Cindy Hunter, who just really infected me with a passion for seaweeds that I feel a great responsibility to pass on and to ensure that this legacy of phycophilia is continued throughout our next generation of young scientists. And so each of my students that graduates gets a Gen Seaweed pressing to make sure that they don't ever forget about the importance and wonderful uses of seaweed, including their artistic applications. And my students and I also are very passionate about ensuring that we communicate our science to the public to make sure we spread our phycophilia to other members of our community. And so we're often engaged in outreach activities in our community here in San Diego and beyond. So with that, I would just like to end by saying that I think seaweeds are California's most vastly underutilized resource in a whole variety of different ways. And I look forward to this festival now and in the future helping to make this no longer the case. And so thank you again for hosting, and happy to answer any questions.
Okay, thank you so much, Jen. That's amazing. I really have to compliment you on your beautifully clean cultures.
Yeah, it's a challenge.
Nice.
It's taken a while.
Okay, so I want to introduce each of the other panelists before we come back for questions, but I can see there are already questions piling up, so rolling in. Okay, our next panelist is Dillon Dolinar. He's a graduate of San Diego State University. His research experiences include studying fish in South Africa and Honduras. He's a scuba instructor and has spent a lot of time in and around the kelp forest ecosystems of Southern California. Dillon is currently the Southern California volunteer coordinator for Reef Check, a citizen scientist organization. Most importantly for me, Mr. Dolinar is currently studying rhodolith beds on Santa Catalina Island here in Los Angeles County. So tell us more about your work, Dillon. Don't forget to unmute your mic.
Alrighty, thank you very much. Here, let me begin the screen sharing process. Make sure that this works okay.
Yes, beautiful.
And then start. All right. So this story begins, like Janet said, on Santa Catalina Island here in Southern California. It is not only the center where the story takes place, but it's also important to me on a personal level because it's really where I gained my love and passion for subtidal ecology and particularly seaweed ecology. For many other people, though, Catalina Island means something different. It means a wonderful place for them to be able to take their very luxurious yachts and being able to visit in that sense. And when all of these yachts are moored up on the mooring chains out here in the mooring fields, when there's swell, we see that the mooring chains that they're tied onto can get lifted up and down like this. And this chain it will create a big disturbance and essentially crush anything that is living on the benthos. And so when we look at places on Catalina Island, what is exactly living on the bottom of these mooring fields? And it turns out they are these really small red coralline free living algae called a rhodolith. And they tend to aggregate together through Santa Catalina Island in multiple places. And they're extremely ecologically important organisms. They are ecological engineers in that they build a large three-dimensional habitat that allows all kinds of wonderful organisms like this little pike blenny right here, and allows them to have a great habitat for them to thrive in. And so we have all kinds of other invertebrates. Here we see about a good size range of the rhodoliths. This is a photo taken by Paul Tompkins, who is really influential in mapping all of the rhodolith beds on Catalina Island. And so we can see that maybe a large one would get around in between 2 to 3 centimeters here in Catalina Island, but they can get bigger elsewhere. Additionally, something that makes them really special is they're one of the few algae that we have a fossil record for. We're able to actually see fossilized rhodoliths, and that is really important in providing more information about them. And so, like I said a little bit, they like to hang out in these sheltered, shallow coves, which right here on Catalina Island, here are a few of in which they aggregate in and make large rhodolith beds. And these are all places that have mooring fields. So they like to hang out in the same place as we like to, in a sense, as well. So a study by Gabriel and others in 2018 found that when the rhodoliths get disturbed by these chains, we see large drops in abundance of everything from macroalgae to invertebrates to even fishes. And so we know that this is affecting the ecology of rhodolith beds, this disturbance. And so what I wanted to do is I wanted to understand what's happening at a physiological level when these rhodolith algae are disturbed. And what we see is that the rhodoliths have a lower respiration rate when they are intact and undisturbed. But once they get smashed by the mooring chains, their overall respiration will increase. So we were curious if this happens immediately or only happens over time. And so we can see here, we ran an experiment where we measured the respiration of the rhodoliths right before, and then we just crushed them with a lead weight and measured their respiration right after. And we see that there was a bit of a little increase right here, but it's not quite up to the higher respiration levels that we saw among the ones that have been out in the field for a while. Additionally, we wanted to look at what happens with primary production of the rhodoliths as they get disturbed. And here we did not really see a quick drop off with our immediate crushing, but we do see that their ability to photosynthesize drastically decreases over time if they are subject to that repetitive anthropogenic disturbance. And so that's only one part of the picture. We wanted to know what's happening to the other members of this ecosystem. And so what we looked at is the respiration rates of 5 prominent invertebrates who inhabit these beds. We have the little cone snail right here, a larger wavy turban snail, and then a few other as well, such as a predatory sea slug like a navinax. And what we see is that they respire in different ways. Not everyone's respiration rate is exactly the same. And we scaled this to the size, the mass of the organism. And basically the reason why this is an important question is if we go back to this, we're losing a lot of respiration, like community respiration, when the abundance of organisms decreases. And so this is important. So we have our wavy turban snail right here. Its abundance almost completely is lost when in the areas where there's the mass disturbance in the mooring fields. And they were our organism with the highest respiration. So essentially the whole community is losing that oxygen intake. And so the main conclusions from what we've been looking at is that we know that the mooring disturbance can decrease the rhodolith production and it'll increase the respiration. But we know that the immediate disturbance can reduce the production, but not necessarily the respiration immediately. And then lastly, when we combine the field abundance and lab-based respiration production rates, we can better model how this mooring disturbance is impacting these subtidal algal communities on Catalina Island. And to go full circle here, as Jana said, I now work for Reef Check California, where I have been doing many kelp forest surveys. And we're looking at the ways that marine protected areas can impact the abundance and densities of marine macroalgae all over the state, and also invertebrates and fish. So I want to thank all of these wonderful people for helping me along this journey. I'm very thankful for each and every one of them, and I'm looking forward to hearing everyone else's presentations.
Okay, thank you so much. I'll see some questions that are directed to you, so we'll get back to those after we hear from everybody. Okay, our next speaker is Gabriel Montecinos Arismendi. He's a graduate of Pontificia Universidad Católica de Chile and has
Don't worry.
has a Master of Science degree from University of Pierre and Marie Curie in Paris. He has expertise in the genetics of brown algae and also more human-centric questions, such as how to develop market potential of seaweeds for food. Currently, Mr. Arismendi is working as research assistant at the University of Wisconsin-Milwaukee, where he is part of a large research group investigating potential breeding programs for the iconic canopy-forming kelps of California. Please tell us more about it, Gabriel.
Okay, so I'll go with the tradition to share my screen. Share here. Share. Okay. So yes, I'm part of the Felipe Alberto team in University of Wisconsin-Milwaukee. So the first interesting thing is we can do in marine science in Wisconsin. So we need to recreate this tiny ocean in the lab. So that was the first challenge. And we can do, and we work in genetics, and especially we try to do this kelp seed bank. I want to explain now. So we're working in genetics. Why genetics is important? So you have here in the example 2 population, and you can see some variability in terms of size. So this red line represents the average size, and you can understand this size as attribute, and this attribute is consequence of this, we can call it equation in between interaction between genotype, environment, and other factors. But controlled environments, maybe it's not impossible, but it's very hard. So we're working in genetics. We try to understand the genetic part of this component of this equation. So actually, you see again the difference in natural population. Many farms working with release spore or take individual from natural population. So what you obtain normally is some attributes very similar to this average natural population. So the idea for us is that to improve this characteristic. So if for that you need pass for 3 stages, this is the process of domestication for seaweeds. First step is characterize the difference in genetics in the natural population. The second step is collect and store individuals with different genetics. And third is cultivate them and try to obtain a specific strain. That is the process, need more selection. So when you pass of these 3 steps, you have this consequence in these 3 factors. So you pass it between step 1 to 3, you want to reduce a little bit the genetic diversity because you select specific genes, specific individuals. Second, you can improve your yield and your income, but for this, you need more research and development. So that's the process what we are now. So the first step I told you before is characterize. In this case, Felipe Alberto Lab has 10-year experience in characterize the genetics in brown seaweeds in California and the West Coast. So in case, for example, Microcystis, we already know we have 3 different genetic groups, so represent for each color. and one mixed zone. So this is the gray color, but at least we have 3 different genetic groups. So potentially 3 different attributes. And the second step of this domestication process is collect and storage. So how we make that? So you go to the field, and what you can see is the sporophyte, this big plant that everyone knows. This plant, these seaweeds, not plants, but this seaweed releases spores, and these spores differentiate in this particular process or different structure called gametophytes. And these gametophytes differentiate in male and females. In that, exactly what we can work. In particular condition, this gametophyte become reproductive and complete this life cycle and obtain a new sporophyte. The interesting part is we can keep these gametophytes. I'm expert in this area, and we can keep them. That's what we call kelp seeds, because potentially we can use for seeding structures or seeding string or for farms. So here is solely, I can show you all the process, what we're doing, starting with releasing spores, after differentiate males and females. And this part you need to believe me. It's hard, but you can believe me. I can identify different sex in this tiny organism. So I can differentiate males and female, grow them, stock them with this D image. I can stock. And after I can grow this biomass of this organism and multiply them continuously I can have kilograms and ton of these individuals I want. But we can use 2 for a specific crosses and obtain a small individual what we can use. So actually we have 4,000 isolated gametophytes from macrocystis, young kelp, narrowcystis, bull kelp, and we can complete the life cycle of these 2 species. This is essential for this domestication process. So we are in this 3rd step of the domestication. Already we are result of our 2nd farm. And you can see some results in this picture in top right. So we select some individuals, maybe 3 feet tall, so small individuals or average individuals. And we have this other result when you have around 8-9 feet individuals, just for a process of selection of these parents. And we make a very complex analysis, genetic analysis. We identify in the genome of this species, you can see this color bar in the bottom left. So each color represents, we can say, a chromosome, and we identify 8 regions associated with the growth rates. So we can select, that's the idea for the first farm, select individual for this. But this project is not only breeding and selection. We can use this seed bank or this gametophyte for recovery populations. And that's the case, for example, of bull kelp. Now we have this new project funding in California for collect more seed bank and collecting more gametophyte. And you can see in this image that it's complex, but in simple, each dot in this image in the California coast and Oregon, I think it's the state, or whatever, in the west, in the Northwest Pacific, each dot represents a population analyzed with 100 individuals, and each color represents different genetic groups. So we already know bull kelp have different genetic groups. And in the left side, you can see the genetic diversity. So other thing we know now is Alaska and California have very rich diversity, and some places like Puget Sound in Seattle have low genetic diversity. So these places have potential important to conserve in case of the California for very rich in genetic diversity, and Puget Sound probably for the low genetic diversity is something we need to conserve. So now just invite everyone to use this gametophyte we have already in the lab. So we have the content here for everyone, and you can use this local kelp seed for restoration, extract, research, and farm. Thank you. No? I can continue talking or... I think I want to explain more things.
Okay, thank you so much, Gabriel. That is fascinating stuff, and I am sure you're going to be taken up on the offer to people to use the genetic stock of bull kelp that you have in Milwaukee, of all places, to replant bull kelp on the California coast. That is a fantastic resource. And more questions coming up. But first, Professor Matt Edwards. Okay, Matt received his PhD from the University of California, Santa Cruz, and he is currently full professor at San Diego State University, where he runs the Kelp Forest Ecology Research Group. There, he has mentored more than 20 graduate students and authored more than 60 publications and book chapters. Matt is an expert in kelp forest ecosystems of the Eastern Pacific, especially the Aleutian Archipelago and California. And through his body of work, you can see running the common thread that the biology of the seaweeds themselves can drive what happens in kelp forest ecosystems. So tell us more about it, Matt.
Thank you very much. As I'm switching over to share my screen, I'll say that so far in this panel, we've had some really nice examples of research that on how we grow seaweeds, why we want to increase their biomass, and what happens when we lose them. So I actually want to take a step back for a second. I want to talk a little more generally about seaweeds and why we might want to study them. I'll say that this really starts back when I was much younger, probably about 25 or 30 years ago, and I had one of my uncles ask me, why do I care about seaweeds? We're probably speaking to a sold audience where most of you chime in like seaweeds already, but still I kept this with me. And as I teach classes in seaweed biology, one of the things that the students, other than having to take a required organismal class, is to say, why do we want to study seaweed? So I want to talk a little bit more generally here and simply present to you, can seaweed save the world? And so I'm going to present kind of general information from other studies and then finish this with some of the work we're doing in my own lab. So one of the things is, for those of us who have thought about terrestrial forests and seaweed forests or kelp forests that we've heard about, right, there's some similarities. Seaweed forests are incredibly important ecologically. They provide habitat and nursery grounds and energy for a whole host of other species. They're economically important, not just for their harvest, but for ecotourism and fisheries, both recreational and commercial fisheries. But getting back to this thing about other than those, I want to pose to you, can seaweeds help cool a warming world? As you heard Dr. Smith talk about a little bit ago, the world's warming, right? And so we can look at this. One of the aspects of why the world's warming are greenhouse gases, as what was mentioned. And we know they're rising. And as those greenhouse gases, the CO2 interacts, it diffuses into the seawater, becomes aqueous CO2, and it decreases the pH of the seawater. So it's not just the atmosphere that's got problems, it's the ocean that has problems too. So what about seaweeds? Well, some of the work I did with some colleagues at Santa Cruz and Alaska Fairbanks, we looked at just how much carbon can be held in a kelp forest. And the reality is you have about 100-fold more carbon locked up in a kelp forest than you would if you lose that kelp forest, say, to Urchin Barren Formation, which we're seeing huge problems in the Northern California and Oregon coast and up to Alaska. If you take a look just how much carbon in this bottom graph here is held, the top graph is held in a kelp forest, we can look at some of the major forests we typically think about, like rainforest and boreal and deciduous forests and evergreens. And kelp forests hold almost as much carbon as those forests do. Primary production on the bottom graph, they actually have higher primary production, higher carbon sequestration. They just turn over a little faster. So they're really important to kind of global carbon cycles. I'm not going to go too much into this because Dr. Smith talked a lot about this, but one of the other things is methane, right? Stronger greenhouse gas than CO2. And Dr. Smith told us about, this red algae called Asparagopsis, and feeding these to cattle in small amounts can help reduce methane production. right? Another thing is we can ask a question, can seaweeds feed a hungry world? Well, we all are probably familiar with nori, or if you've had sushi, it's a red alga that is really important. It's one of the world's largest seaweed industries. It's just one example. Lots of food comes from them. There's other species that have products like carrageenan, which is a thickening agent. And if you go to the store and you look at things like, oh, here, just even look at, Ben & Jerry's ice cream or Twinkies, and you look at the what goes into these things, you'll generally will see something like carrageenan or some other product from algae that's in these things. So algae can be really important in feeding, as a food source, right? In other areas of the world, seaweed farming is hugely important. These are seaweed farms just in Korea. I was over there looking outside my window, and that's a large barge. You can see these big square seaweed farms where they're farming these things for food production. This is in British Columbia. You can see here a kelp farm up in British Columbia. And I'm all the way over on the left, a long time ago when my hair was a little less gray. Another thing that we can talk about is, can seaweeds help reduce the world's trash? Well, actually, some of the seaweeds, particularly the brown algae, have alginic acid that we can extract and take the compounds out of them and use them as a thickening agent. And, we think about just plastic straws. We all should be aware of this, that, reducing our reliance on plastic straws, on plastic in the environment. You can make plastic straws and cookware and eatware out of seaweed products. Another thing we can do is, we can build things that are slow release agents for things like Argentine ant control. This is a group out of UC Riverside that's been able to embed things like boric acid, which is an ant control agent, into this. And you can put this out in the environment and you're not putting plastic out in the environment. But taking all that together, we can see that seaweeds can have these big purposes. I want to talk a little bit about some research in my lab that we're doing that's asking if seaweeds can remove excess nutrients from estuarine waterways. So we should be aware that nitrogen, phosphorus pollution from industry, from agricultural practices, from residential activities, and there's this, right? These things can put a lot of nitrogen and phosphorus into the environment. And when it rains, what happens? This stuff heads to the storm drains and eventually makes its way to the sea. It's really important for a place like, say, San Diego Bay that sits next to almost 1.5 million people, with a lot of nitrogen and phosphorus going into the ocean, in addition to organic pollutants. So with colleagues from Sunken Seaweed in the Port of San Diego, some of you may have seen Tori Polizzi and Leslie Bohr from Sunken Seaweed earlier this week, talking from the farm at the Grape Street Pier in San Diego. There's a farm established there where we're growing seaweeds on the farm, mostly Tori and Leslie are, and growing different seaweeds, things like sea lettuce, ulva, or giant kelp that we've heard about, macrocystis, and some red algae like gracilaria. And we wanted to know if these things could be grown on these farms during these rain events when fresh water is washing these nutrients to the ocean, but then the sea's becoming less saline. And so as this goes on, we're going to look at if these seaweed farms can even be used during this time. Can the seaweeds grow in these, they're ocean algae, but can they grow in low saline environments? So we can ask how decreased salinity and increased nutrients uptake ulva's physiology, its nitrogen and phosphorus uptake abilities. Can it be farmed? So we do things like we'll bring the seaweeds into the lab and we'll grow them under different salinities, say typical ocean salinities around 35 PSU, and cut it in half to around 15. And then we can add different amounts of, say, a commercial fertilizer, really high, medium, and low amounts. And then we can look at how ulva grows and how it survives in its metabolism. And so we do this in a number of ways in my lab. I'm not going too much. We can measure things like growth, which is a standard one that you've heard a lot about today. We can also look at their photophysiology, how well, they're photosynthetic organisms. There's a photo part of it where they're uptaking light and they're capturing that light and turning it into energy. And then there's a synthesis part of it that they're taking that energy and they're fixing CO2 into sugars. And so we can look at the different aspects of that. We can also look at their ability to take up things like carbon, nitrogen, and phosphorus. And welcome to Zoom. There goes my dog at the door. And And then we can look at carbon and nitrogen assimilation pathways using stable isotope analysis. And so that's one way of looking at how these things, and I'll tell you right now that Ulva does very well under low salinity conditions if you give them nutrients. It's an ideal candidate for bioremediation, for cleaning the San Diego Bay of Water. And we're going to try to expand this to different areas of the bay. Question is, I'll kind of wrap this up by saying, what about other uses of seaweeds? Again, we can extract things from their thickeners. We can extract things like potassium, phosphorus, and nitrogen from them. And by the way, during World War I, we actually used kelp to make cordite and gunpowder when there was an embargo against bringing these things in from Germany. Anybody's been to the dentist and had to bite into one of those things that's all mushy and they take your dental impression? That's from kelp. And it's alginates. You know, I always heard about, you know, if there's a problem with the nuclear radiation, say we had the Fukushima Daiichi nuclear disaster, and someone might say, you know, we saw this, it released a lot of radiation to the atmosphere, and someone said, take a pill. And I'm like, really? What kind of a pill can do this? Well, kelp contains iodine 127. And if you saturate it, it can saturate your thyroid, it can prevent the thyroid from taking up radioactive forms of things of iodine and strontium and cesium. So it can actually protect you from nuclear radiation. And so I'm going to just end this thing by saying, yeah, I totally believe that seaweeds can save the world. We've talked about food, we've talked about greenhouse gases, but there's a whole host of other things that seaweeds can do for us. And, you know, it's an untapped resource and something that I think we need to spend a lot more energy in developing our seaweed cultivation techniques. So I'm really interested to see the stuff that Gabriel and my colleague Felipe are coming up with about growing seaweeds, these special strains, and things that Jennifer Smith is coming up with about, you know, increasing our production of these things to reduce methane production. So I'll leave that with you that, you know, if I were to answer my uncle now, I'd say, well, the reason why I study seaweeds is they're going to save the planet. But you can help everyone.
All right, cool.
Forms. Also, watch what you throw away because there actually is no away. So seaweeds can help, but you got to do your part.
I agree completely, Matt. I really think that, you know, we need to work with the seaweeds to sort this stuff out. That the seaweeds are almost like growing behind us to clean up after what we're doing in some ways. So we can work together with them to make things better in the future. There are so many questions. So I'm really excited to get going with questions. So starting with Dillon, there were some questions. I'm going to kind of roll several questions together. Are the rhodoliths protected, and why are the mooring chains made of something so damaging? And how did you get so much work done while you were an undergrad?
So to answer the first question about their protection, so many of these of our sites across the island were actually in marine protected areas. Like the first one to come to mind, if any of you have been to the Wrigley Station at Big Fisherman Cove, right next to Two Harbors, that area is a marine protected area, and there are rhodoliths there under that mooring field. And so I think that from that standpoint, they were able to like, it's almost like the policymakers are probably trying their best. Like I don't really know the best way to get around that, which kind of feeds into your second question, which is about why are they so damaging. It's because the boats that they're holding on cost a lot of money. And if the material was a lot weaker, then we could have multi-million dollar yachts breaking off of the moorings and then hitting the island. And that's a whole nother kind of person that'll be upset. So it's really trying to, now it's going from the what's happening to like what we're talking about with the what can we do about it, which is the next process is working with engineers to find ways around this problem. And that's the step that we're beginning now, which we're really excited about. And then lastly, for the undergraduate, I guess just never saying no to anything. If someone says like, do you want to work on this thing? I'll be like, yes. Like I'll get 6 hours of sleep, but I'll do it. And I think that that's a really important thing, is that opportunities seem infinite, but they're very finite, and you have to really work with what you're given. And then you can use that to mold what you hope to be like an accomplishment down the road.
Oh, thank you so much for those comprehensive and insightful replies. The next group of questions molded together again is for Jennifer, and it is Asparagopsis the only seaweed that reduces methane production, and why? Well, how much are farmers committed to using this, and why would they be interested in it?
Yeah, great set of questions. So there's a bioactive compound that a variety of different seaweeds, actually many seaweeds produce, called bromoform, which is shown to be the active compound at mitigating methane production in ruminant animals. And it just turns out that Asparagopsis is the stellar performer in terms of bromoform production. So it produces, an order of magnitude, sometimes more bromoform than other seaweeds. And so while we might be able to use other seaweeds for this solution, you would have to feed them, the ruminant animals, a lot more biomass to achieve the same results. And in fact, even the trials that have looked at other seaweeds have never been able to get close to the values that have been achieved with Asparagopsis. So that's a great question, but at this point in time, Asparagopsis seems to be the star performer. There may be other seaweeds out there that we have yet to discover, and there are a lot of groups around the world that are looking into this. Obviously, this is a global problem that could have star seaweeds in any variety of parts of the planet. So we're not expecting to solve this problem all by ourselves here in California either. In terms of farmers' willingness to use the supplement, I think it varies widely depending on where you are. Obviously, there's not a lot of desire to have more costly supplements going into this industry. And so finding ways where you might get mutual benefits. So it's been shown that Asparagopsis actually the supplement makes cows more food efficient. So they actually digest more of their food, which then means you theoretically would have to feed cows less to achieve the same health. And if that's true, there might be just a cost zeroing out for the supplement, makes them more efficient so they don't have to eat as much. But there are big incentives to addressing this issue. First of all, the livestock industry gets a really bad rap from the sustainability perspective. And so they are interested in improving their sustainability. And in places like California, we have a new law that by 2030, all livestock farmers have to reduce their methane output by 40%. And there currently is no real viable solution for doing this, aside from Asparagopsis, assuming we can get it on the market by 2030. We got a decade, so fingers crossed.
Okay, great. Yes, all of us are doing that, right? Okay, the next two linked questions are for Gabriel. And they are there any organizations already using your kelp for restoration? And has any other culture previously tried to domesticate kelp?
Okay, so they're very good questions. So first, yes, we start working with Puget Sound Restoration Funding in Puget Sound, and already we already setting all these part with send samples and everything going well. So the pilot works, and next year we want to put more individuals with particular crosses, so particular individuals, and test they survive better or what happened with them. So yeah, we have an institution working there, and next year we want to prove better this concept to restoration with Neurocystis vocal. In terms of domestication, in US, no, in my knowledge, no teams tried domestication. Many groups working with traditional ways, sporulate or release spores into string, just seeding them. the traditional way. So working with this stage with gametophyte, the seed bank, not yet. So that's the essential part for domestication. In Chile, you have some work, but that doesn't include the genetic part, what we're doing now in this project. So the classic domestication is going to take many years, using genetic tools, maybe 5 years. We are in this process already.
Okay. Mm-hmm. All right. There are a number of questions about
Sorry, I just forget one thing. Forget just this part of the project. What I show, we're working with Pete Raimondi, San Francisco University, Santa Cruz, yes. So we collaborate with him, and they send us samples to try to keep more gametophyte from California area, and potentially help them to use, because they have a project funding for understanding, try to recovery bulk kelp. It's a huge problem in California. 90%, if I don't wrong, 90% of the bulk kelp disappear. So
yes, in some places, and then other places less. Yes.
Yeah.
Yeah.
So we are part of this project, helping with keep gametophyte here.
It's wonderful. It really is. There are a lot of questions. You can all just jump in as you feel you can about effects of various kinds of pollutants on seaweeds. And the kinds of pollutants that people want to know if they affect kelp forests are radiation and microplastic and pH changes related to CO2 pollution. The radiation one was specifically asking about like if there were other big sources of radiation, like other accidents.
I guess I'll start in for the group. You guys can jump in. As far as radiation goes, kelps are great sentinels. They're great, really cheap kind of canaries in the coal mine. They can tell a lot about what's happening with radiation. There was a big project called Kelp Watch that was collected kelps from pretty much the entire Pacific Rim following the Fukushima Daiichi disaster, seeing if we can trace radiation in the kelps, because they're great sponges. That's one thing. They're tremendous sponges for whatever's in the environment. They'll take it up. And so, I haven't seen evidence of negative impacts to the kelp themselves. The levels that we were detecting are very background. Getting an X-ray, not even that, but they are detectable. CO2, that's a whole one. That's something that my lab does, and we do look at ocean acidification and ocean warming. And you warm the ocean, and algae, most of the kelps we have do poorly. They don't like warm water, especially because it comes with low nutrients. But if you warm the water and you give them excess CO2, there's actually some sort of a physiological uptick in them. You kind of are increasing their metabolic needs, and you're giving them more carbon in the water. And so while the kelps may do a little bit better physiologically, I want to leave it that it's not a good thing, because there's so many things that come with that warming ocean. Storms, which are the big dog as far as taking out the kelp forests. Those kinds of things, the kelps are sensitive to a lot of these. And I've spoken enough. I'll let some of my colleagues speak about the things they've done.
I'll add a few things. Oh, sorry. Seaweeds are an amazing form of bioremediation, as Matt talked about. They actually essentially act like sponges, and they take up whatever's in the water column. And so if it's heavy metal, pollutants, toxins, whatever, in some cases they might be negatively affected, but in the most cases, they're going to take them up and store them in their tissues, which means that we can then take that biomass out of the water and use it for restoration and for enhancing water quality. So in some ways, seaweeds are this amazing source of, they have this amazing ecosystem service, as Matt talked about, for being able to clean the water. I'll just add a little thing too with ocean acidification. It has been shown, we've done a number of experiments as well as a whole variety of other labs, but it seems to be pretty consistent that the seaweeds that produce calcium carbonate, the corallines that Dillon talked about, and other calcified species tend to be more negatively affected by ocean acidification than the fleshy algae, which often can actually increase their photosynthesis. And so we need to be cautious of that, especially because some of these corallines have really important ecosystem services, such as acting as settlement surfaces for a whole variety of invertebrates. So there could be these cascading consequences associated with the different responses to climate change impacts, such as OA.
Yeah, absolutely. And to build off that, once you factor in temperature, then we're getting a whole mess, because then, like Dr. Smith just said, they might not do the best in the OA when the water is becoming more acidic. But if you give them warmer ocean temperatures, there was a study out of South Korea that looked, and they talked a little bit about they looked at how they respond just under increased ocean acidification, and they found that production and growth decreased. And then looked at what happens with the temperature. They said it went up a little bit. And then when they combined the model, what we're going to get with the warmer ocean and the more acidic ocean, it almost sort of wiggled out in a plateau. And so it's so much fun, because the answer in ecology is so often sometimes and maybe. And so this is a good example of how there's so many things happening right now that to isolate them is really good, and putting them together is a mess, but that's what we do.
So yeah, that is why we need to do more science, right? Is that the answer is not always what the answer was somewhere else, right? So it's very
from one species to the next,
right? Or from one species.
As Dr. Smith said, the things that have calcium carbonate, I mean, it's, imagine dropping a little bit of acid onto some coralline, some calcium carbonate, it'll dissolve it. There's a problem with the acidification side of things. But also one thing, without going too far down this rabbit hole, as the ocean becomes more acidic, the carbon dioxide that goes into the water actually changes form. It becomes bicarbonate. Just think about your carbonated soft drinks. It becomes that, but also there's a carbonate, and the carbonate is calcium carbonate, things that build skeletons, your coral reefs, your coralline algae, sequester that out of the water. As the oceans become more acidic, that carbonate actually becomes less and less and less abundant. It starts actually becoming bicarbonate as well. So the ocean acidification is harming them just by being more acidic, but it's also making it more energetically costly to grow.
That's right. I find the same thing in some other species, just like you say. Same thing, slightly different, balance in every system. So we have a question about, I wanted to go back about microplastics. Anyone know about microplastics in seaweeds?
We haven't studied that in my lab, so I can't speak to that one.
I mean, the great thing is that seaweeds don't have mouths, and so they can't ingest plastic, which is, the biggest impact in most marine organisms. So I would say, luckily for us, seaweeds are not negatively impacted broadly by microplastics. Unless the plastics are causing a reduction in light and their ability to access light. When you get those big rafts of plastic growing or drifting in on the surface, that could have a negative impact. But other than that.
Yeah, the only way I think they can affect, for example, plastic is sometimes plastic includes some component of heavy metal in the production. So that probably heavy metals or minerals, metals in particular, could be affected more than just the particle of plastic. So I agree with Jennifer. I don't know the negative impact from kelps for plastic already.
I have heard similar thing. One thing to consider in the plastic, that the plastic is, as it's breaking down, is releasing so many different chemicals that is very hard to tell what the effects are, because it depends on what plastic, like whether they are full of metals or not, and other chemicals that are in there, and organic molecules that might affect the microbiome on the surface of the seaweeds. So yes, lots of different things that come.
More science. We need more science to understand that.
So much better. So that gives me a tiny segue to something that's coming up on Friday on Sustainable Tech Day, as one of our panelists makes seaweed plastic in California. So come back then to learn more about alternatives to plastic. Okay, here's a question about macrocystis. Is macrocystis everywhere the same species?
So this is a good question. It's a
loaded question.
But I can respond in terms of genetics. So the studies show macrocystis is one single group in the entire world, including South Hemisphere, in Argentina, Chile, Argentina, New Zealand, South Africa, and some population in Ireland. The same group. This is the genetic concept of species. In the biological concept of species, where you need to cross individuals and have fertile individuals or next generation, they continue doing the same species. But in terms of ecological difference, they have some difference in some particular structure. So the holdfast, for example, in some species is larger, and we're talking about morphotype integrifolia and morphotype pyrifera, because they have maybe some particular difference to try to be better prepared for this environment. So macrocystis, in my knowledge, is one single species. You have probably, because it's not ready, I don't see any papers say that yet, but probably have ecotypes independent of the environment where they grow.
Yeah, I would add to that if that question had popped up 5 or 10 years ago, the answer would have been completely different. Just not that long ago, we considered them different.
It's the new tools finally, the new tools show something different now. Genetics show something. Before it's more basis in morphology, what we call now ecotypes. And so before we base everything in morphotypes or morphology. So in based on morphology, they have some difference, but in genetics and crosses, it's the same species already.
Fascinating. Okay. I think I'm gonna kind of wrap up the questions soon. Oh, related to that question was a question about how the kelp forest in different places affects the flow of water. So does it protect the coastline? Does it make it safer on the coastline? So this is really a forest level question, I think.
Yeah.
Matt.
Yes. Easy answer to that is yes. And this has been known actually for quite some time now, going back into the 60s, that, the kelp forests dampen both kind of along shore, the currents that come down the shore, they slow them down. So the edge of the kelp forest has a very different current regime than it does in the center. Also wave attenuation. That's being studied with this kelp forest in the North Coast with some collaborators from UC Davis, putting out current wave meters and things that measure water motion and looking at as the kelp forest to recover, how does that affect it? But yeah, absolutely. Kelp forests, their physical structure, they do drag, they do slow down the water.
All the people with the big houses really appreciate the kelp forests when they can overlook them and are protecting all of that.
Okay. I think all the people who go to the beach appreciate it too. Through the public access between the big houses. Okay. Let's see. All right. I have a question, a general question for everybody. And I'd like to have everybody kind of weigh in, which is, how can people, this is also, aimed at you, Dillon. How can people who are not going to graduate school and getting a graduate degree in marine biology, how can just people get involved in science about sea?
That's an excellent question, because the grad school route is not for everyone. It's kind of funny because I will be going that route, but I'm not there yet. I haven't taken that exit. And so this is a little bit of a plug for the organization I work with. It comes to mind really well.
That's what I was going for.
California. So what Reef Check California does is we sort of specialize in a citizen science, where we take people who might not have the formal marine ecology education that we have, but we teach them standardized surveying techniques, and they get to take really good quality data to look at what's happening along the California coastline. And our biggest thing that we're looking at right now is comparing what's happening in marine protected areas and what's happening in not marine protected areas just outside. We're seeing, is there a difference in algal abundance? Is there a difference even in maybe fish size? Because the fish are being fished out of the non-MPAs and they are protected inside. And so with that, someone who we welcome everyone. And so we have people who are lawyers, who are doctors, and we also have marine ecologists who just on the weekend, they want to go diving and take some fun data. So I highly encourage anyone interested in doing subtitle surveying to check out Reef Check. And also just, I think that it's fun. I don't know, it's just because I'm a big nerd, but to read things, like it's a little old fashioned, but to read papers and to read textbooks teaches you things that you didn't even know existed. And so that's, if they're nerdy enough, a path I would recommend down that road too.
Okay. Anyone else want to weigh in on how people can get involved with science?
So I'd recommend just participating in ONGs because, for example, in Puget Sound, they have a very nice program in there. See people, volunteers to participate in surveys, sometimes in kayaks, in sea species. It just take notes about present, absent of some individuals. So organization like that helps you to have information. So yes.
Yeah, there's a whole variety of citizen science organizations in California that can get you involved in collecting those types of data. But also, if you are near a university, you can always reach out to faculty members and see if they have opportunities for volunteering. We've had a really diverse group of volunteers in my lab over the years, ranging from retired, engineers that just want to get their hands wet to high school students. And, if you have graduate students that could use an extra set of hands, it's really nice to have volunteers. And so there are groups at universities that'll allow you to experience science without actually having to get the degree yourself. And yeah, last but not least, I'd say, read. There's a ton of great information out there, and some of the older literature, even though it might be a little outdated, is incredibly important for all of us to kind of ground ourselves in where this field came from to better understand where we're going.
Maybe I'll end on this one. So this is a question I get asked a lot. I am the Marine Emphasis Coordinator. I oversee the Marine Biology program at San Diego State. And I get asked a lot, how do I get involved? And Dillon, actually, this is how I met Dillon some time ago when he was a student. He came and looking for things to do, and he was traveling the world, doing things, and he got involved in research in our lab. But more to people who aren't going to college, I would say knock on doors or send emails, I guess is the current way of saying that. We're not knocking on doors right now, but send emails to people. You'd be surprised. I know it seems a little bit daunting, and it seems like, oh, I don't want to bother this person, or I'm afraid to do it. Don't be. As faculty members, and people who run labs, we take a lot of people. As Dr. Smith will tell you, our labs, we have a lot of people that come and volunteer from us, from high school to students to the general public to university students. So don't be afraid to do it. Look at web pages, send an email saying, hey, I'm interested. Do you have opportunities to volunteer in your lab? I think you'll be surprised at how many people will actually say yes.
I agree completely that, we all want to share what we do with more people. That's why we're here. I have one more question that came in early, and I kind of missed it at the beginning. So I don't want to lose this one. It's about red tides. Remember, we have sometimes we have really serious red tides here in California. How does that affect the rest of the seaweed? Does it affect seaweed farming? Does it decrease the biodiversity of seaweeds?
Yes. Does anyone? Well, I've spoken the last one, so I'd let somebody else jump in if they want to.
No, go on.
Okay. One thing that happens with red tides, it's not so much, the red tide, it's the organism itself, although for the things that ingest it certainly can be, because they do have toxins associated with them. But something that happens with red tides is they change the chemistry of the water. And, you can make the water go anoxic. We think of these things as being photosynthetic organisms that are producing oxygen. But when you get really high abundances, actually, it can go the other way. These things, they're also organisms. Plants produce oxygen, but plants are also respiring, as Dillon's research showed. They got to get rid of waste. They got to grow. They got to, deal with damage. They've got to reproduce. And so these things can really suck down the oxygen, and that can be a huge problem. It can take up the nutrients and compete with other algae. So red tides themselves actually can be problematic. They also happen to occur at a time when the water's warm and the nutrients are already kind of poor. That's one of the telltale signs of a red tide. So they kind of go hand in hand. But yeah, the red tide itself can certainly have strong impacts on algal communities.
Yeah, during the last big red tide that we had here in San Diego, we measured essentially anoxic condition off the Scripps Pier for a period of 3 to 4 days, which is essentially, zero oxygen in the water column, which is why we had not just, effects on the seaweeds themselves, but many invertebrates and fish, washing ashore dead. And on top of that, the bloom was so thick that it literally shut out the light. And so a lot of the seaweeds on the bottom in the kelp, whether it be the kelp themselves or the understory, were just starved of light for extended periods of time. So while the red tide organism itself might not directly affect seaweeds, it's this phenomenon that happens associated with the blooms that is kind of an ecosystem-wide response.
Okay, so really that ties together a lot of what we've been saying about how the seaweeds are in the ecosystem and partly running the ecosystem, but responding to everything in it. So when something else blooms, then the seaweeds, have to make room for that. So I want to kind of wrap up, but because thank you so much to all our panelists for coming and telling us about your work and putting it out there in the California Seaweed Festival and supporting our event, and for sticking around extra time to answer all these questions. I also want to thank all the people who've been writing questions in the chat. We haven't been able to exactly answer every single one of them, but we really do have to kind of wrap it up now. But you can find the contact information for all of our panelists on the California Seaweed Festival website. So you can ask them more questions there, because you can send them an email like Matt just told you to. So it's just, has been a fabulous conversation. I'm so glad that we had you all here. Okay, I want to make a few announcements before we go. Remember that we screen videos at noon, Thursday and Friday, and 3 p.m. at Saturday. Oh, maybe 1 o'clock on Thursday. And everything's 12 o'clock. Okay, Thursday and Friday at 12 o'clock and 3 p.m. on Saturday. Okay, if you missed anything from the California Seaweed Festival, it's archived on our YouTube channel. So the live events are there for you to watch later or to share with your friends. I want to thank especially our live stream producer, Elizabeth Burns, who, with technical assistance from Tim Aguirre and Steve Dudgeon helping out in the live stream chat, have made this possible. Julia Marsh of Sway provided the graphic design. Today's events were sponsored by California Sea Grant, CSUN, Yukon, the Cultured Abalone Farm, Sway, Sunken Seaweed, and Nature Spirit Herbs. And okay.
All right. Thank you, Janet.
Bye-bye.
Thank you.
Bye.
Good.